Good morning, everybody. And I'm Tomoaki Ikeda, obstetricians from Japan. Today, I would like to discuss fetal heart rate variability. And this is the outline, my talk. The first, I will discuss the physiological origin of the fetal heart rate variability. The second, I will review evidence from anencephaly and hypoxic ischemic injury. Finally, I will introduce our recent genetic studies focusing on the NALCN gene. Among all CTG parameters, I believe baseline variability is the most important. The major CTG parameters include baseline heart rate, variability, acceleration, deceleration, and uterine contraction. Variability is defined as irregular fluctuations in fetal heart rate occurring at least twice per minute without regularity and in amplitude or frequency. Clinically, variability is classified absent, reduced, moderate, increased, or sinusoidal. Why is the variability important? Because it is one of the best indicators of fetal acid-base status. My mentor, Paula, reviewed multiple studies and demonstrated that when baseline variability is normal, the probability of significant fetal acidemia is very low. In contrast, when variability is absent or reduced in association with decelerations, the risk of acidemia increased substantially to 23%. Therefore, variability provides important information about fetal physiological reserve. What generates variability? Variability is actually a mixture of oscillatory systems operating at different frequencies. At approximately 0.05 Hz, there is intrinsic vascular rhythmic activity. At about approximately 0.15 Hz, oscillations arise from baroreceptors or chemoreceptor feedback loops. At higher frequencies, around 0.5 to 1 Hz, fetal breathing movement contributes additional fluctuations. Therefore, baseline variability is not a single phenomenon, but a result of several physiological oscillators interacting with each other. This concept can be illustrated by the sinusoidal pattern. In the 1980s, we performed experimental in fetal sheep. After parasympathetic blockade with atropine, we infused arginine vasopressin, which enhanced the intrinsic vascular oscillations. As a result, sinusoidal heart rate pattern was generated in sheep. Interestingly, arterial blood pressure simultaneously showed sinusoidal oscillatory pattern known as Meyer waves. This suggested the sinusoidal fetal heart pattern originate from the oscillations in blood pressure regulation. In summary, baseline variability represents the physiological complexity emerging from multiple biological systems. These include vascular oscillations, fetal breathing movements, fetal activity, baroreceptor reflexes and chemoreceptor reflexes. Therefore, variability should be viewed as a physiological signal rather than the simple noise. I will now move to the second part of my presentation. What happens to variability when the fetal brain is injured? A key study addressing this question was performed by Terao in Japan in 1980. They investigated CTG finding in anencephalic fetus. Their result demonstrated that when medulla oblongata was absent, baseline variability disappeared. Because the medulla contains a major autonomic cardiovascular control centers, this finding suggests that the intact brainstem is essential for generating variability. This concept is also consistent with clinical observation in the cerebral palsy. Dr. Nakao said the last session. Some fetuses already show absent or markedly reduced variability at admission or at the onset of labor. These cases are classified as the NR pattern, non-reassuring pattern. The likely explanation is that hypoxic ischemic injury occurred before labor and caused brainstem dysfunction. This raises an important question. How long does variability remain abnormal after hypoxic injury? So to answer this question, we use the fetal sheep model. In this chronic preparation model, fetus is temporarily removed from the uterus, electrodes and the vascular catheters implanted, and the fetus is then returned to the uterus. After recovery, controlled umbilical cord occlusion can be performed to induce hypoxic ischemic stress. This model allows continuous physiological monitoring under a near normal condition. To quantify variability objectively, we use the two standard heart rate variability indices. RMSSD reflects parasympathetic activity. SDNL reflects overall autonomic nervous system activity. These parameters allow quantitative assessment rather than subjective visual interpretation. We compared the sham controls with 15 minutes and occlusion and 25 minutes umbilical cord occlusion, 3 groups. Immediately after occlusion, both RMSSD and SDNN decreased. In the 15 minutes group, mild type, variability recovered within several hours. However, in the 25 minutes group, a severe case, variability transiently increased, then decreased markedly. This reduction persisted for more than 72 hours after hypoxic ischemic stress. Histological examination confirmed severe brain injury in this group. Therefore, prolonged suppression of variability appears to reflect neurological damage. We then next followed these animals for 21 days as an unexpected finding emerged. Variability showed a clear circadian rhythm. Values were higher during the evening, lower during the morning. Evening value recovered within approximately 5 to 7 days after hypoxic ischemic injury. However, morning value remained suppressed even after 21 days. This indicates prolonged impairment of autonomic regulation. We then examined whether a similar phenomenon exists in humans. Using 231 severe cerebral palsy cases, we compared variability during the morning and evening. The results closely matched those observed in sheep. Variability was significantly lower in the morning than the evening. Thus, the circadian modulation of variability appeared to be present across species. I will now move to the third part of my talk. Can genetic disorders also influence fetal heart rate variability? This fetus was referred to at 30 weeks because of decreased fetal movement. The CTG showed almost complete absence of variability. Because there was no evidence of acute hypoxia or maternal drug exposure, we suspected an underlying neurological disorder. 1 week later, variability remained absent. However, acceleration associated with the fetal movement was still present. This unusual pattern suggested preserved movement but impaired neural regulation of variability. The infant was delivered at 34 weeks by cesarean. He demonstrated distal arthrogryposis, hypotonia, and abnormal limb posture. EEG, electroencephalography, performed at 21 days after of the live, showed complete absence of REM sleep. Genetic analysis identified Clifford syndrome caused by NALCN, narcin mutation. Clifford syndrome was abbreviation of congenital contractures of the limbs and the face, hypotonia, and developmental delay. Previous studies by Funato in Japan showed the narcin dysfunction markedly reduced REM sleep in mice. This finding immediately attracted our attention because REM sleep is closely associated with fetal heart rate variability increase. So narcin is a sodium leak channel that regulates neuronal resting membrane potential. Changes in narcin activity alter neuronal excitability and influence REM sleep regulation. Therefore, narcin provides a plausible biological mechanism linking neuronal activity, REM sleep, and also fetal heart rate variability. To further investigate this hypothesis, we generate narcin is a sodium peak. To further investigate, this is a heartbeat in the zebrafish. To further investigate this hypothesis, we generated a narcin knockout zebrafish. Compared with wild-type animals, knockout zebrafish demonstrated markedly reduced heart rate variability. Both SDNN and RMSSD was significantly decreased in the narcin knockout zebrafish. These findings suggest that narcin plays a fundamental role in the generation of physiological variability. Finally, baseline variability is not noise. It is a physiological signal generated by the multiple interacting biological systems. Medulla and brainstem are essential for maintaining variability. Persistent loss of variability reflects the brain injury and autonomic dysfunction. Genes such as narcin and mechanisms related to the REM sleep contribute directly to variability generation. Therefore, baseline variability provides a unique window into fetal brain function. As AI-based fetal monitoring advanced, I believe the next challenge is understanding the physiology behind those patterns. Thank you for attention.
Thank you very much for an interesting talk. So, please, questions? I see one there.
Thank you so much. Excuse me. That was so interesting and highlighting, as you do, it's the complexity of the different physiological signals. Is we can't just look at one and not pay attention to another rhythm, for example, and the systems that are associated with those. So I'm wondering, maybe this is too specific a question, but it strikes me you had or do have a unique way to also look at the effects of respiration on your heart rate variability. When you're the zebrafish, I'm guessing, there isn't any respiration, so you can disassociate respiratory activity in order to understand your RMSSD changes. In your other experiments and the other work you've done, have you been able to look more closely at how respiration affects your heart rate variability, particularly RMSSD? And as you know, that probably is very different in REM sleep versus non-REM sleep. That's a long question, but
yes,
have you been able to look at respiration?
Because normal respiratory rhythm is 0.5 to 1.0 hertz. But we sometimes observed is abnormal breathing after the hypoxemic injury. It's a kind of the continue 15 hours, something like that. So this respiratory rate is more slow, and maybe 0.5, something like that. So we should differentiate is normal fetal breathing meant from the abnormal fetal breathing meant. And recently, my colleague and Chris Lear had observed even in the normal during labor, the fetus abnormal breathing is occurred after the contraction. Yeah, this is a...
Yeah, and the problem, as you know, they don't breathe all the time. Being able to know when they are breathing and when they aren't is such a thorny issue. So thank you.
Thank you.
More questions or comments to Professor Ikeda? Yes?
Thank you for your talk. It was interesting to see the circadian variability in your patients. I was wondering if you'd looked at circadian variability under normal births.
That's a good question. That he is now doing that normal. Yeah, we'll talk.
So I'll come back in 2 years and you'll have the answer.
Yeah. In sheep, the normal sheep is very shallow circadian rhythm. But sheep with brain damage is so exaggerated circadian rhythm. Now he is doing observing the same things that happen to the human.
In the sheep who were injured as a result of the prolonged umbilical cord compression, and you followed them out the evolution. Did at any time in the evolution you challenge them with even a brief period of hypoxia or asphyxia? Because if the responsiveness is altered, it is going to likely alter the response to even small provocations. And so that is another part of the information you're getting.
Thank you. Yeah.
Other question, comments?
Just comment on behalf of Chris Lear. So there are two aspects for this study, Barry, where you're picking up now, I don't think they challenged afterwards these fetuses, but we did study, and they studied some of the chronically hypoxic lambs that are spontaneously chronically hypoxic, and then we would see when they do the occlusions on them afterwards, how quickly they deteriorate compared to normally normally non-hypoxic lambs. So that's one. But the experiment in which you cause brain injury by prolonged occlusion, then let it recover a couple of days and then start hitting, I'm not sure they did that yet. Do you know, Professor Ikeda?
No,
they have, but it's definitely something that should happen. These are very difficult studies to do. And one thing I wanted to say as well is I think the idea of this a lot is that when you look at variability, the clinical message here is as well, when you look at variability, and when you say, oh, it's reduced, oh, it's increased, there is more to it. That's, I think, the clinical message. There is more to it. And sometimes other movements, other things are masking the reduced variability, which is signaling to you that that baby is already vulnerable or damaged, or it may not be damaged. It's not doing its normal cycling. But because there is things like sinusoidal is one of the great examples that if it looks sinusoidal, you would say, oh, it's normal variability. No. And then there could be breathing movement, there could be other, and we've seen so many patterns in which the variability is reduced, but there are other signals, whether breathing movements or so, they are masking that reduction. So the clinical message is that, you know, our simplistic way of going reduced, absent, you know, it's good, it's a great start, but there's more to it. Sorry for the long ramble.
We had many indices of the present variabilities. Yeah. Thank you.
Okay, I'm looking for raised hands. Thank you once more. So we actually have 5 more minutes to discuss in the whole workshop group. If you sit on any more various comments on the previous speakers also you want to bring up again, since I was so eager to keep time. So, yes, please.
I wanted to pick up on the question, I think it was Philip that asked the question around whether it's surprising to see a discrepancy in the depth of decelerations with fetuses that are exposed to chorioamnionitis. Yes, I wasn't surprised with that finding myself, not least because part of the problem of chorioamnionitis obviously is fetal host response to infection with output of inflammatory or pro-inflammatory mediators. Now, many of those mediators modify and modulate cardiovascular regulation, cardiovascular system regulation. And some of it also feeds into attenuation of the depth of decelerations or response to hypoxic ischemic insult. We see exactly the same picture in fetuses that are exposed to chronic hypoxia. When they develop tachycardia, their decelerations are also shallow because their responses have become blunted. And because we know that those pro-inflammatory mediators also interfere with the ability of the fetus to respond to hypoxic ischemic insult, it's not surprising, therefore, that in chorioamnionitis you do have shallow deceleration instead of deeper decelerations.
Yeah, I find it also logic when you think about it. Do you want to comment, please, Taro?
No, I agree closely in everything that he said. So.
Any other comments?
Okay, I'll do one more. Sorry, and then we can go to topic. But I think that's, again, to try to summarize what we're talking about. If you think about deceleration as a healthy response of a healthy fetus to the contraction, good healthy deceleration, then when we see the chronic hypoxic, what Austin mentioned, when now we see the choreo, just who knows, a few other scenarios are there as well. These babies, you just can't respond with that healthy deceleration. It can be shallower, maybe not shallow always, but shallower, maybe not as rapid. So there's increasing evidence across the board. So Austin, would you agree? I think that's, I'm trying to kind of, again, bring it in a common language what that means.
The problem is there are so many different inputs even before we begin to monitor the babies. So, for example, I am more comfortable about a baby that came into labor with a normal CTG and then subsequently became tachycardic and mom developed fever and so on and so forth, compared to a baby that is admitted with those CTG changes already in place. So there's also, I suspect, a time-dependent effect on some of the observations that we make on CTG. If the exposure to infection and chorioamnionitis is fairly recent, then the time of exposure, obviously depending on the virulence of the organism, is shorter compared to a baby that that is admitted already tachycardic, already decelerating in terms of what how much reserve that baby has. So I personally think there are so many variables that it becomes really difficult to tease out what. Absolutely. Tachycardia was mentioned, but I know for sure that there are no specific CTG patterns of chorioamnionitis. It could be anything, and tachycardia is just one of it.
Okay, thank you all for a very interesting start of all our sessions. And we go for a coffee break now until